Thank you very much, the organizer, for inviting me. So far, you are listening from the clinicians. I think I'm the only basic scientist, and I'll be talking about the gut neuroscience, nervous system in the gut. Okay. But so far, since the morning, we just heard from the clinicians. Okay. So you can see a picture that sometimes we think it is just maybe the animation, but this is not an animation. This is how the brain, how the neurons talk each other. They're exchanging some ions, and this is one of the ions, like the calcium. When they communicate each other, this calcium ion goes in and out. That is the recording. It looks like an animation, but this is the real picture we take. So human body has a second brain that is called enteric nervous system that controls the movement of the food throughout the digestive tract. And this enteric nervous system, it lies entirely in the wall of the gut, beginning in the esophagus. So I can. Yeah, very good.
The next slide.
No. I'm just turning give it to me. So starting from the esophagus through all the way to the rectum, this nervous system exists. So you can see the enteric neurons. These neurons are actually clustered or grouped in ganglion, and these ganglia are connected through the bundle of nerve fibers, and they are actually they actually form two types of plexi. One is called myenteric plexus, and other is called submucosal plexus. The myenteric plexus, these neurons are actually in between the longitudinal muscle layer and the circular muscle layer, and it is throughout the gut. And you have submucosal plexus that reside in the submucosa, and it is in some parts of the gut missing of the submucosa because the submucosal functions are mostly in the absorption and the blood flow, these type of things, and you don't need that type of function in the stomach, so you don't have the submucosal plexus, those neurons in the stomach. So these myenteric plexus neurons or submucosal plexus neurons, they can work independently without any intervention from the central nervous system. You see, there is a video on the bottom. This is the gut, particularly the distal, the colon, collected from a mouse, and it is in the organ bath, and you can see it can contract and dilate without any influence from the central nervous system. And the total number of neurons are more than 500 millions in the gut. That is pretty much nearly same number as in your spinal cord. So the myenteric plexus neurons, they are mostly control the gut motility, the contraction, relaxation, how your food or the contents move from all the way from oral cavity towards the rectum. That is controlled by the myenteric neurons. You have the submucosal plexus, as I mentioned earlier, that mostly control the secretion and the vasodilation. So as I mentioned, that enteric neurons, they can function independently, but still the central nervous system can modulate this function. So central nervous systems are connected with the enteric nervous system through its two branches, sympathetic nervous system and parasympathetic nervous systems. For the sympathetic, the cell bodies are in the paravertebral ganglia next to the spinal cord. They are projecting all over the gut, from the esophagus to all the way to the rectum, not esophagus, starting from the stomach to all the way to the rectum. For your parasympathetic, you have got the vagus nerve that is mostly in the stomach and the intestine and proximal colon. The vagus nerve does not go to the distal colon, but the the central innervation comes through the pelvic neurons. They innervate the rectum. So this is the external innervation to the gut. As I mentioned earlier, the enteric neurons that's in the gut, there are different types of neurons there. Some are called like the sensory neurons. When you eat something, the gut, what is called the gut, feel the mechanical tension. The sensory afferent neuron, they send information to the motor neurons. They can contract, relax, to propel, to move the food content or the waste material to get out of the body. These are the different types of neurons. So the smooth muscle cells, the force of contractility for the bile is actually provided by the smooth muscle cells. They actually coordinate very well to mix the food content and to move the digestive undigested material from the body. So these smooth muscles, like the motility, their movement is, you need different types of pattern. It's not like only the contraction or relaxation, but this contraction relaxation should be very organized. It is coming from the excitatory motor neurons. From the enteric nervous system, you have mostly excitatory and inhibitory motor neurons. The excitatory motor neurons, they send their projection usually towards the oral, the proximal, and the inhibitory motor neurons, they project to the distal, to the inner side. So if you consider a specific region, at the simultaneously, the excitatory motor neurons, they are sending projection to the oral side that cause contraction. In the bottom part, you need dilation. That's the inhibitory motor neurons, they cause that dilation. So the food can mix, food can move forward. That type of pattern is called peristalsis. But along with the peristalsis, you also need segmentation, migration motor complex. All these different patterns are coming from the smooth muscle cells, and different types of neuronal intervention cause this type of pattern. And you see, for your heart, you have got, like for your blood or circulatory system, you have a strong pump. Heart is pumping out the blood throughout the body, but your gut, you don't have such pump. The enteric neurons, they are mostly controlling these very organized movement. Like in some portion, there is contraction, relaxation, all these things. So this is the excitatory neurotransmission, how the neurons talk to the smooth muscle cells. Neurons, they release the neurotransmitter. And that neurotransmitters bind not only with the smooth muscle cells, there are other supportive cells. They are called ICC, intestinal cell of Cajal, and also like the fibroblast type cells. That neurotransmitter bind with the receptors sitting on the smooth muscle cells and the other cells. That's how they communicate. They cause the contraction. And the inhibitory motor neurons, the neurotransmitter, mainly nitric oxide, and also the VIP, PACAP, that type of neuropeptides, they also help to cause the relaxation. So I'm not going to details about this busy slide. It is mostly the cellular mechanism, how the smooth muscle cells contract. It is mostly due to the calcium ion goes in, they bind with the calmodulin. That's a protein, that calcium calmodulin binding causes some second messenger mechanism system to cause the contraction relaxation. I'm not going to that details. So how I'm actually different lab. We are like when we have been approached with these mice, the smooth muscle problem. So very first thing you see from the outside, they have a big belly, a stroke or the vasculopathy, you cannot see from the outside, but these mice acted to mutant mice. From the outside, you can see they have a big belly. When we open it up, we can see they have the small bowel from the stomach to all the way. This big thing is called cecum. It looks very normal, but after the cecum or including the cecum to all the way to the colon, they looks very dilated. You see the fecal pellet on the left panel. I'm saying this is the control mouse. I'm saying all the way healthy, and the mutant mean your ACTA2 mutant. I'm not going details about the ACTA2 because Dr. Lindsay and all the speakers touched base about this mouse model. So you see from the cecum, this side, this is the mutant mice. You don't see any fecal content shape. They are just like the mesh type things. They are like swelling up here. And at some point, you see there is no further movement of the content after this point. So that may be the reason we believe of the cause of this mouse death, like the brain stroke and other thing. I think the prominent cause for this, particularly for these mice, may be the obstruction of the bowel, the fecal pellet, or the fecal content cannot get out of the body at some point. We measure the size, that's very easy. We can see there is significant difference of the colon size. The colon is shorter in these mice. Then we wanted to see actually what happens to the bowel movement, whether the bowel can move, they can contract, relax or not. So this is one of the same video you saw at the beginning. I need your help, might be. Okay.
Go back to the presentation.
Beautiful, thank you. So this is a 10 minutes video, which is compressed in 1.5 minutes. If you count the number of contraction, you see how the contraction goes from this side is the oral, and that's the rectum side. You see it's moving all the way very beautiful. If you count this contraction, I'm sure you can count 6 times it's happening here. And from this video, we make a map that's called a spatiotemporal map. That is bottom here. You can count that number of contraction. At the same time, we measure the luminal pressure. We put a catheter in the inside of the gut, and when the contraction happens, as the fluid cannot go out because we make a tie on the both sides, pressure builds up, and we can measure the pressure. We call it luminal pressure. You see, every time the helpful contraction happens, we see the luminal pressure goes up. And that's how we count the number of contraction, and we can also measure the velocity. But when we come to the mutant mice, you see the difference. You can see there is the vibration. The muscle, smooth muscle cell, they may try to contract, relax, but they can't. You see the vibration. And from the video, from the map, you cannot see any such like the band things. There's the contraction that we call colonic migration motor complex that is absent in these mutant mice. And look at the pressure. They have very minimum luminal pressure, but not that type of peak to cause the contraction. But so far, whatever the mutant mice, that one of the mutant mice has no contraction at all. But at the same time, we have another mouse that has got different types of motility. If you look at this pattern, sorry.
You can probably just go like this.
Okay. Have a look here. That is very slow.
Normally
and you
have
can
the lab meeting using because insert floor. And yes, I think it's better than the sandwich downstairs.
neurons to release the neurotransmitter, electrical field stimulation. What's happening here?
That's a good question.
Okay. So after we give the electrical stimulus, all the neurons release the neurotransmitter, particularly the excitatory acetylcholine that bind with the receptor sitting on the smooth muscle cells, and they give a big contraction. Here you see the big spike, that's a big contraction. That happens, that's very normal, that's in the healthy. But when you look at the mutant, you see they don't have that abrupt peristalsis or that thing. They have got very rhythmic contraction. That is the muscle, smooth muscle phenotype. If there is no neuronal intervention, muscle can contract by itself all the time. That is very rhythmic. From our experience, we know it. And you see, this is very rhythmic because the neurons are not actually interfering with their work, with their function to do. But when we give the electrical stimulus, the neurons should release the neurotransmitter and they should give a big spike of contraction. But that did not happen with the mutant mice. So we don't know actually, is it the neuronal problem or the problems with the smooth muscle cells? This is very slow. Sorry about the technical issue. I don't have any more video. I think it should be fine now.
Okay.
So many files are open. I'll get there. Yeah, beautiful.
Yeah, put it back to place.
I don't want anymore. Maybe that's fine. Yeah, that's okay. Then what we wanted to see if the neurons are actually cannot work, if they cannot release the neurotransmitter. So we given the acetylcholine, that's the excitatory neurotransmitter from the outside, to see whether that neurotransmitter can talk to the smooth muscle cells. You see, with the healthy, as soon as we give the acetylcholine, the number of contraction goes high. You see it here. But in these mutant mice, that sick mice, somehow they lost even their smooth muscle, the normal contractility by themselves. So we believe the neuron can maybe okay, but the neurotransmitter, what should bind with the receptor on top of the smooth muscle cells, maybe something wrong there. That's why we can't see any function after the neurotransmitter adding to the bath. Then we also added the potassium chloride that called global depolarization, cause the muscle to contract. That can be a huge contraction. That's the normal phenomena. Even we check the viability of the smooth muscles. Adding this drug, we see in the healthy, there is a big contraction, but in that mutant mice, the contraction, even their normal contraction, they lost that contraction. So definitely something wrong with the smooth muscle. Then we know from our lab, we published a paper a few years back. It was showing that you need the enteric neurons and the smooth muscles. They actually feed each other, they crosstalk each other. So you need both components to get a beautiful function. But from the developmental perspective, if you don't have the enteric neurons, we have seen the smooth muscle cells can still grow very well. But if you take the smooth muscles out of the gut, the neuronal pattern particularly lost. Because at the beginning, I told you the myenteric plexus, they're actually in between two muscle layers, in between longitudinal and the circular muscle. So if that muscle cells are missing, they cannot give a pattern of the neurons. So we thought maybe as the smooth muscle are the issue here, so look at the total number of neurons. So we looked at the total number of neurons. They're exactly same. They're healthy. The shape, morphology looks very good. There is nothing wrong with the enteric neurons of these mutant mice. So it confirms that the problem can only be in the smooth muscles. So sometimes these, when we saw these dilated gut, we think that can be maybe inflammation, maybe other stuff is going on. So we looked at the inflammation marker and we did the histology. We don't see any abnormality in these gut with the comparing with the mutant. Then we also did the immune cell infiltration that gives you the message that whether the immune cells, particularly the leukocytes, they should be outside the gut, they should be on the luminal surface, on the epithelium. But when you have the inflammation, that immune cells go all the way to the enteric neurons. So that is called like the infiltration, but in the healthy or the mutant mice, we don't see any immune cell infiltration. So from the inflammation perspective, we don't believe that these mice have any inflammation in the gut. So it's only about the smooth muscles. So that's how we characterize these mice, shows all the gut phenotype, all the sick phenotype. Then we wanted to see whether we can help with any medication. So Dr. Mussolino, Dr. Lindsay, they have beautiful group. They are working on different types of gene editing therapy, different types of already FDA approved molecules to cure this gut phenomena. So I tried just one of the drugs. This is the drug treatment on the right side. So you see that look at that the morphology or the structure. It looks very normal on the treatment side. You see the fecal pellet. You see the fecal pellet has got its own shape. What was missing in the mutant. So the colon size again, the size was shorter in the mutant, but we can restore the normal size of the colon after giving this treatment. That's the first thing. Second, we looked at the survival curve, like whether they survive better or their body weight gain is improved. So this work was done by Dr. Vijay, Dr. Sabia. It is their work. They looked at these parameters. They see this treatment improves actually their survivability and also the body weight gain. They also checked something called rotarod. They put the mice in the what is called like the treadmill type thing, and that treadmill is not like the horizontal, it is moving around, and the mice control the what is called control, and they cannot stay there longer, particularly the mutant mice. They fall from that treadmill, but after the treatment, you see they can stay there longer as like the healthy control. Then I came back to my work, like to the smooth muscle contractility, whether we can restore that function. Again, we put that treated mice in the bath. We give the electrical stimulus. This time we can see the neurotransmitter. They can talk to the smooth muscle clearly, and we can restore that smooth muscle contraction. Then we also looked at the acetylcholine, what is the main excitatory neurotransmitter in the gut. We added that thing from the outside, and that caused the function, normal function was missing in the mutant mice, but we can restore that in the treatment group. And again, the potassium chloride, the global depolarizing chemical, that also restored the function in the treated mice. So that's all from my side. And I'm part of the Goldstein lab. I would like to thank Dr. Mussolino and her group, and also Dr. Lindsay, to give us the opportunity to work with them. And it's a fantastic opportunity for me to listen from the patients, to see, and it's a motivation for the basic scientists. We feel better to work actually more, to put our full dedication to get something out for you guys to help the patients to cure the disease. Thank you for your attention. I'm happy to take questions, maybe not very related with the clinical. Yes, please.
So with your research, is there a chance, for the people that have the complete gut failure or the intestinal failure, whatever, is there a chance that you can reverse that?
Yes, like at least we tried few of the medications, but one of the medication what I am presenting here, I don't know about the other organs, but for the gut, we see it is improving. They're living longer, and their like the gut function is better. When I did not do any like the full motility, yet. I'll do it in future. But so far, the morphology wise, the length, the size of the fecal pellet, this everything is revived, is becoming normal in these treated mice. Hope for the best.
Ahmed,
Yes.
do you mind if I add to that one?
Sure.
So we treated the mouse so far in very early time points. They're baby mouse when we start treating them. We are now treating older mice. To your question, once things are not working for a long time, do we gain anything? So far, we do see an effect. Is not as robust as what you're seeing. So the better is the sooner we can get to this system as it's being developed and it moves and it learns, and babies start with milk and then we go with solid food and the gut is learning with them too. So we know that any treatment for the disease will have probably a higher impact in the symptoms if you can start it earlier in the disease than later. And that applies usually to all diseases in general in humans.
Let me preface this by saying I'm not a geneticist, so I will try to be as clear as I can. So when we heard in the earlier presentation about the NR treatment, and they were talking about its impact on the alpha-actin protein, and then there was some discussion about its impact on the gamma alpha protein in the gastroenterology. Can you talk a little bit about that? Have you tried or looked at those mechanisms specifically?
I can say only one thing: the gamma actin is also expressed in the smooth muscle cells in the gut. So we did not look at yet. So as we know, the problem with the alpha actin, alpha smooth muscles. So we did some immunohistochemistry to see the leveling. And we did not see the gamma actin yet, but it is already in the literature that gamma actin is present in the gut. And if we see that there is like the expression is less or more, then we can comment about that. We did not do the expression of gamma actin yet, but I'm hoping to do it in future. Thank you. Yes.
Hello. I don't know anything about gamma actin in the gut.
But that is,
I saw that what Dr. Murdoch presented. So what does that protein mean to you, that other actin isoform? Is it needed for function?
Isoform, I don't know the functional implication, but there are very, very few literatures coming out about the gamma actin in the gut.
Okay.
Yeah.
Okay. Thank you. I'm just trying to understand.
No, it's not out of blue. The work is there. Gamma actin is present in the gut.
Okay.
Yeah. It would.
No, I'm learning too. Ahmed's been teaching us about the gut, and it's just not something. I mean, there's just different specialists for different organs, and we're all trying to learn about it.
Yeah, we all are trying to learn from each other. That's the beauty of science. Nothing is hiding. Nothing is hidden.
And I want to call out and thank. There is another scientist that works with Mark Lindsay's laboratory and Dr. Raj, who kind of shares bench with these guys. That's how they connected. And then when they knew that you guys were in, they took us on. So I just want to transmit that the people that you brought to this room, the drug that is being used, we cannot fully disclose it, but it comes from a collaboration with an Israeli group that was here also participating 4 years ago. And so we kept taking what they were doing and seeing if we can try it in the mouse, and very preliminary stuff, but we've never seen anything that can make the gut move. It's a lot of the smooth muscle, a lot. So these are, at least we are in the right direction. It sounds like it will be hopefully available. It's FDA approved. We don't need to go through making it from scratch like gene therapy. Okay. So for you guys in timeline, this is why we're doing these things. This is why Dr. Milowicz is also rushing, because we want something now to make people better. And whether we can help the gut, we can help the vessels, we can help the aorta, we'll find out. We still need all the way.
Oops. I am the grandfather of Parker Scott. So until this comes into play, are there things that we can do with the grandson, like smaller meals, probiotics, prebiotics? What are the type of things that we should be doing as grandparents? No sugar, for sure. Stay away from the gluten. We understand those things, but that gut-brain connection is so huge. What can we do?
It is hugely impacted. Like they're now a very new field in the science, that gut-brain interaction. Your mood is also, your food is not good, your mood is not good. Many things happening. So far, like the gut can, like I think, like for this on problem can be the blood flow that is missing from our work in the gut. We are just looking how the gut is functioning. The smooth muscles, maybe you also need the blood flow in the gut as well, as the brain and other part of the body is suffering from the vasoconstriction or less blood flow. That can be another reason in the gut that can cause all these problems as well. So,
And for the what do we do? We have Dr. Moran part 2 on Saturday. So if you guys can hold it until then, I think we're going to have more experienced gastroenterologists, just for the clinical side.
wait for the right
I don't want to
question.
throw you on
Personally,
the ring.
yeah,
with, like,
that's fine.
how do you feed the baby? Thank you. Thank you so much.
No worries. Thank you.